not allow the fusion. This is anti-fusion drug. It is anti-romance drug. Yes, what is this drug? This drug is called enfuvirtide. 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 Right? Again, this is also available in the market. Right? We are very rapidly progressing to understand this virus. Right? So up to now you have learned, we have only talked about outer two molecules and we have talked about two drugs. Maraviroc, which does not allow the GP120 to bind with CCR5 and does not allow proper attachment. Then other drug is, first was attachment inhibitor, other is fusion inhibitor, which bind with GP41 and does not allow it to work. And name of this drug is enfuvirtide. Okay? That's right. Okay, now let's enter in the cell. Sorry, this is not a cell. This is a virus. Okay. Now we see what is inside. Another thing before I really go inside, this protein, these two proteins, GP120 and GP41, they are present on the envelope. They are also called envelope protein. What are they? Envelope protein. And the gene, there is a gene, the gene by which these are, the gene which expresses and make these two proteins are initially make that single unit. That single unit is called GP160 and then it is divided into two components, GP41 and GP120. This gene which makes these envelope proteins, what is the name of this gene? What are the spellings of envelope? It is ENV. ENV. From NV, not only it steals the, what is this? Clothing. It has a beautiful jewelry, deadly. Girls have these jewelries. So this is make everyone envious. So what is this gene? ENV gene. So ENV gene is responsible to produce envelope glycoproteins. These are two. Am I clear up to now? Now I think we should enter inside the virus, inside the envelope. Inside the envelope, this is the place where true viral genetic material lives. This is the house. And this house is also made of, yes, not glycoproteins, only proteins. Glycoproteins are proteins with sugar, sweet proteins. The sweetness is shown outside, right? Not inside. Okay, inside is just proteins. Okay, so glycoproteins are on the surface, they are not inside. So this is the real house of the viral genome. Viral genome and important enzymes are here. Is that right? Outside this was just a bubble, and on the bubble these were glycoproteins. But there is one thing. So that actually to stabilize these, virus makes another protein which is present under this area, right? And this protein is like this. This is what do you see? What is it? What is it? It is M, of course, not any other thing. So this is M. These proteins which are just inside the envelope and they basically stabilize these GP41 here. These proteins are called M for matrix. So matrix proteins. What are these called? Matrix protein. So these were glycoproteins or envelope protein. Inside the envelope, what are the proteins? Matrix protein. And matrix proteins are called P17. Actually, these numbers are given according to their molecular weight, right? That is why they are very random type of numbers, right? This is also called P17. So P17 is just inside the envelope, and this makes what? Matrix protein. Is that right? But I'm not going to spend time on that, thank God, because it's not so much clinically important. We come to inside this structure. This is actually capsid. What is it? Capsid. And this capsid, right? This capsid has how many sides? 20 sides. 20, as you know, a few things that this is triangle, this has 3 sides, this has 4 sides. Is that right? This actually structure, it has how many sides? 20 faces, 20 sides. So because it has 20 sides, it is called icosahedral. What is it called? icosahedral. So capsid is, under the electron microscope, it looks like a cone shaped structure, but actually it is icosahedral. Icosahedral. Okay? So this is icosahedral. Now this house in which the RNA of the virus lives, this is also made of proteins, right? The proteins which make this structure, these proteins are P24. Yes, keep on saying, what is it? P24. P24, yes. Say loudly, what happened? P24. Now, this has a lot of clinical importance. P24 is important. Good doctors should know what is the importance of P24. Number one, very important thing. P24 is not involved in attachment and fusion. Is it involved? No. So if we make antibody against it, now listen very carefully. If our immune system make antibody against P24, and actually it does, the antibodies which are against P24, can it block the entry of virus into next cell? No. Because entry is dependent on GP120, not into P24, right? Because once primary attachment, secondary attachment and fusion occur, this whole structure is thrown into target cell. Is that right? When this membrane, envelope membrane and membrane of what? Target cell, they fuse. Then through that, when the fusion occurs, a hole is produced. Through this hole, this viral capsid is thrown into target cell. So, if we have antibody against it, it is not important because still virus will enter into target cell. So, the antibodies which are anti-P24, are they neutralizing or non-neutralizing antibodies? Non-neutralizing antibodies. This is one thing you should know, right? That the antibodies against P124, only P24 are non-neutralizing. Secondly, this protein is relatively stable. This protein is relatively stable. What I mean by relatively stable, that it does not undergo antigenic variation. But it doesn't matter to us. We really want this to be stable, so we can neutralize it. But unfortunately, this is not stable. This keep on changing its phase, and this is stable. Right? But what is the importance of this protein? Another very important thing is about this protein. That is about diagnosis of this virus. Yes, please, Jamshed.
That's what I was
You wanted to tell this. Okay, tell me, please.
Sir, it is used in diagnosis.
Yes?
Diagnosis of HIV, AIDS, it is used.
Very good. He is trying to tell us that this protein has some clinical importance. Again, let me tell you, you only learn basic to practice it clinically, right? Now, P24, why to remember this? The reason being that, number one, when a virus enters into your body, you also make antibodies against P24. What are those antibodies called? Anti-P24. They are what antibodies? Anti-P24. Now, anti-P24s appear in our blood after a few weeks of entry of the virus. And now, you know, in a patient, when to detect that HIV is present in his blood or not, usually there are two tests which are done commonly. Number one is ELISA test. What is this? ELISA test. And other is Western blot. These two tests I will discuss into detail later when I will talk about the diagnosis of disease. But what I want to tell right now, that in the ELISA test and Western blot test, we check that patient blood has antibody directed against P24 or not, especially in ELISA. P24, and even in Western blot, we check that patient blood has antibody against the P24 and other proteins also. We'll talk that later. So what I want to say, P24 has a very important thing. What is that importance? That this protein is a very important in serological markers, that usually the people, when we suspect that they may have, for example, if someone get unprotected sex, then he or she is worried that she or he is HIV positive or not, one way is after a few weeks, you take the blood sample and in the blood, they check that person has anti-P24 or not, right? If anti-P24 is present, you think virus may be present. And then we do more specific test, which is Western blot, in which we check antibodies in patient serum against multiple HIV proteins, right? So this is one serological importance of P24. Secondly, another importance. Now we have test by which we can directly check the presence of the protein in the blood. Again, let me repeat it. If I get HIV virus, in my blood, P24 may be present, this antigen may be present, and antibody against this may also be present, right? So it means I may have P24 and I may also have anti-P24 antibody. Am I right? Now in ELISA test or in Western blot, we check that my blood is having anti-P24 antibodies or not, or some other viral protein antibodies or not. Or you can directly check in my blood that P24 protein is present or not. So due to this reason, this structure or this P24 is very important. This is called capsid protein, right? Or this is also called group specific protein, and this protein is serological marker of the infection, this protein itself or antibodies developing against this protein. Am I clear? After that, we enter inside the virus, deeper inside the virus, right? Inside the virus, now you will find, yes, what is this? RNA. This virus this is having RNA as genetic material, and it has double copy of RNA. It has 2 copies of RNA, and both copies are identical. What are these? Every virus has 2 copies of RNA, and both copies are identical. Why I am stressing that they are identical? Listen very carefully. Sometimes, don't call it, sometimes there are 2 RNA molecules, but they are complementary to each other. Complementary mean if here is adenine, then other molecule should have uracil or thiamine? Uracil, right? Okay. If it is having cytosine, other should have guanine. It is not the case in this virus. Both of them are not complementary to each other. Both RNAs are identical. So they are not called complementary copies of they are called identical copies. They are dimers. What are they called? Dimers, right? So we can say that this virus has double copy of RNA, and both RNAs are positive polarity. What are they? Positive polarity. Now you must be thinking, what is positive polarity and negative polarity of RNA? Let me tell you. All of you know that in our genetic material, from DNA we make RNA. And if we talk about messenger RNA, let me tell you, suppose this is my DNA in a gene, and from this DNA you make RNA. Normal RNA, if it is going to make a protein, we will call it messenger RNA. Now I am talking about my genetic material. This messenger RNA, or same as I think yours, messenger RNA structure has one head and one poly A tail. You know that? Poly A tail. Now, from this messenger RNA, you can directly make the proteins. So it means this messenger RNA can be directly translated into proteins. Is that right? Such RNA molecules, now listen very carefully, such RNA molecules which have which are structurally similar to our messenger RNA, that is, they are having head and they are having a poly A tail, and the structure is somewhat like our messenger RNA, right? Such RNAs are called positive polarity. Some of the virus, I'm not talking about HIV, generally speaking, some of the virus have RNA, and these RNA are very much similar with our RNA, messenger RNA, and sometimes they can be directly translated into proteins. So these are called positive sense RNA or positive polarity RNA. Sometimes it happens there is other RNA which is complementary to positive. Now if this is the complementary to positive, can it directly produce proteins? messenger RNA? No. First it has to convert into complementary copy. So such RNA which is not similar to our messenger RNA, it does not have a head and tail, and such RNA which cannot be directly making proteins or cannot be translated well, this is called negative sense RNA. Am I clear? After you have the concept of negative sense RNA and positive sense RNA, or negative polarity RNA and positive polarity RNA, we come back to our HIV discussion that HIV capsid has 2 RNA molecules and both of them are positive sense RNA. But I will make it clear, even though this positive sense RNA which are identical and they are double copy, even though they are like our messenger RNA, but they don't directly translate into protein, I will tell you later there is something unique about it, that from this DNA, from this RNA we are going to make DNA. I mean, it's a little complicated, you know, that RNA of this virus, when it enter into our cell, this RNA does not translate directly into protein. This RNA first convert into DNA. Is that right? First convert into DNA. Now, let's go to another basic concept. Normally, what happens that, let us suppose this is double-stranded DNA molecule, right? And from this DNA molecule, we are making, what is this? RNA. Now, in our body, most of the genes, from DNA we make which structures? RNA. The enzyme which will read our DNA, the enzyme which will read our DNA and make the polymer of RNA, these enzymes in our body, they are called DNA. Okay, let's make it more clear. DNA dependent, because they read DNA. So they are DNA dependent RNA polymerase. I will repeat. If an enzyme can read the DNA and make RNA, we call it DNA dependent RNA polymerase. Is that right? But this virus has a reverse function. What it is doing? That it need an enzyme which will read the RNA and make DNA. It is the reverse function. Normal transcription or normal transcribing mechanism is DNA to RNA. But in this virus, what is there? RNA to DNA, right? So this is also, RNA is being transcribed into DNA, it's a reverse direction. So that enzyme should be called RNA dependent DNA polymerase. As this is called DNA dependent RNA polymerase, then if there is an enzyme which can read the RNA and synthesize, polymerize DNA molecule, that is called RNA dependent DNA polymerase. This RNA dependent DNA polymerase is an unusual enzyme because it is doing reverse transcription, and this enzyme is also called reverse transcriptase. What is it called? Reverse transcriptase. So, because this virus is not so commonly available everywhere, so virus will bring this enzyme with itself. Right? So this virus has this enzyme, and this enzyme is, it is reverse direction of R, reverse transcriptase. Actually, rest in peace. There are 3 enzymes here, and we can remember them by rest in peace. What does it mean, rest in peace? Right? Someone dies and then rest in peace. This is what virus bring the message to you. Now you rest in peace, even though actually we become hyper after knowing that this rest in peace message is there. This R stand for which enzyme? Reverse transcriptase. Right? So I have written it in reverse direction. So this enzyme is reverse transcriptase. Virus brings this enzyme with it. Right? Other enzyme, once virus make the from RNA, when virus make the DNA, viral DNA is integrated in our DNA, and once virus DNA is integrated into our DNA, that enzyme which does this function is called integrase. Rest in peace, another step. Integrase. right? And then there is another enzyme present over there, and when virus make its protein, when virus enter in our cell, and then virus start making proteins for the progeny virus or daughter, or I don't know, this is a daughter virus or son virus, whatever, or grandson virus, the new generations of virus, when for new generations of virus is making proteins, it makes very long proteins which are cut into pieces. And when long proteins are cut into smaller pieces, the smaller pieces of proteins are the functionally active. Is that right? These are functionally active. So it means larger proteins need to be cut into smaller proteins. Is that right? And for that purpose, virus bring its own protease, the cutter. So virus is bringing 3 enzymes. Number 1, how from RNA it will make DNA. Then after making the DNA, how to integrate its DNA into our cell genetic material. And after that, when viral polypeptides are made, polyproteins are made, how to cut them down. What is this? Protease. Why these steps are so important to understand? Because later on I will explain, there are now drugs which can inhibit this. They are called reverse transcriptase inhibitor drug. Reverse transcriptase inhibitor drugs. Then we are also having a drug which can inhibit the integrase, and of course these days we are having drugs which can inhibit the proteases. You are getting it? So that is why we must know this viral structure very clearly. I will repeat it fastly, that basically this is RNA reverse transcribing virus or retrovirus, right? And this virus can be HIV-1 virus, yes, here it is worth mentioning, and HIV-2 virus. HIV-1 virus is the most common all over the world. HIV-2 virus is limited to the Western Africa. HIV-1 and HIV-2 virus are not too much similar. They have only 40% homology. 40% of their genetic material is same, 40% of their protein the same. HIV-1 and HIV-2. But what I want you to remember, this lecture is mainly about HIV-1, which is producing the disease all over the world. But in West Africa, disease is dominantly produced by HIV-2. So this is global, this is West African mostly, this is highly transmissible, and this is less transmissible. That is why it is localized, right? But we are mostly talking about what? HIV-1. Now here I want to mention one more thing, that in the virus now we are having 2 RNA molecules, we have 3 enzymes present over there, we have capsid protein, and there are some additional proteins also inside, and they are called P7, right? P7. These are called nucleocapsid protein because these are attached with this material, right? So nucleocapsid proteins are number 1 P7, number 2 is P24. But which is more important for a doctor? P24, because it's a marker of viral presence in our body, and antibodies against this also tell us virus is there. Is that right? Again I will repeat it. We had the drugs which inhibit the attachment. We had the drugs, how they inhibit the attachment? By blocking CCR5. What was the name of the drug? Maraviroc. Then there was a drug which was not allowing the fusion, inhibiting GP41. What is the name of this drug? Yes? Enfuvirtide. Then there are drugs which inhibit, this I will go in detail in second part of the